All right, so this will be the first lecture into the nervous system. We'll get in this part one, we'll get into the basics of the nervous system, the divisions and so forth. We'll look at the cells that make up it, and then we'll focus on the neuron. We'll look at what's called the resting membrane potential and start into, so remember the neuron does electrical signaling, but then at the end, at the synapse, it's going to transfer that into a chemical signal when it talks to the other cells. So the second part of the lecture, the nervous system lecture, we'll finish up that electrical signaling. We'll look into the gory details of that in part two, and then finish it up with what's happening at the synapse with the neurotransmitter release and so forth. All right, so let's begin our jaunt, and so we'll head into and look at the branches here of the nervous system. We got two main branches. We got the central nervous system that has the brain with all that integration. It's going to pick up the information, integrate it, and it's able to send out a response along with the spinal cord. Okay. We have the peripheral nervous system, right? And the peripheral nervous system can be categorized as afferent, efferent branches, or synonym is sensory for afferent and motor for efferent. But afferent is going to be the sensory. It's going to take information from the peripheral and bring it in towards the central nervous system. So coming from the peripheral back towards the central nervous system, the efferent or motor can take information from the central nervous system and send it out to the peripheral, out to the organs in the body there. Okay, we will spend a chapter looking at sensory, looking at all the sensory organs and so forth. So we'll be looking at these receptors that pick up information, and then the sensory or efferent neurons can bring the information into the central nervous system. Okay. The brain can make a determination of what's in the sensory and can send out a response if we need to through the efferent or motor pathway, okay, or motor nervous system through the efferent neurons that are going from the central nervous system out to the periphery. Now this efferent pathway can be broken down into the autonomic nervous system and the somatic motor system. Okay, the involuntary nervous system, the autonomic, because we don't have to think, we don't have to consciously put thought into operating these neurons. The body does it automatically. So it's all these things like taking care of your blood pressure, raising or lowering your heart rate. These are all taken part by signals sent through the autonomic neurons. Okay, that can be further branched into the sympathetic and parasympathetic. And just like the sensory, we will be spending a chapter looking at the autonomic nervous system. You see, it is very important, and that it is going to be highly involved in that homeostasis process, because it's controlling many of the organs through the body to help maintain homeostasis. Okay, the autonomic neurons are going to control stuff like the cardiac muscle, smooth muscle, exocrine gland, and so all the things we don't have to think about operating, the autonomic nervous system takes care of. Okay. On the other end, the somatic motor neurons. We look at these when we look at muscle contraction, but basically their targets of these neurons are going to be the skeletal muscle. So they control, it's called voluntary because I have to physically or consciously think about contracting my muscles. Okay, so they are controlling the skeletal muscle. Okay, so those are the big branches. We got the central nervous system, peripheral nervous system. We got this afferent or sensory bringing information towards the central nervous system, and the efferent is taking information from the central nervous system and sending it out to the different organs throughout the body. Okay, and so this is how we're going to get our integration. So here's just kind of repeat what I said on the previous slide, looking at the different branches and the organization of the nervous system. We got the central nervous system, right? The brain and the spinal cord, and basically it's going to be there for integrating. It's receiving the information and then it can send out a response. The brain's going to integrate, big center, integrate some of the reflexes. Sometimes that doesn't even go through the brain, it may just go through the spinal cord. Okay, but we're integrating through the central nervous system. Okay, we got the afferent. That's bringing the information in, and again, we're going to spend a good time on looking at how the sensory information. So we're able to pick up information, bring it to the central nervous system. The central nervous system is going to give us our integration, and then the central nervous system can send out information via the efferent neurons, depending on what we need to take place. Say we pick up, hey, we're walking and we see a bear. All right, what do we want to do? Well, you're not supposed to run, but most people will run. The brain can integrate that, hey, I see a bear, and send out to your skeletal muscles, let's get our bodies moving. Okay. The unconscious part, the autonomic division, there is sensory information that is coming from within the body, telling us what's going on. One of those would be like our baroreceptors, telling us about blood pressure, right? And then the autonomic, the central nervous system can send out information through the autonomic division to either raise or lower heart rate, depending on what blood pressure is, whether high or low. Okay, so again, that's going to control smooth muscle, cardiac muscles, exocrine gland, endocrine glands. Okay, all those things we don't have to think about is what the autonomic vision. Neurons are, their targets are those things that we don't have to consciously think about. So let's dive down a little bit closer and look at the actual cells that make up this branches of the nervous system. So we'll look at the neural tissue and two basic types of cells. We got the neurons, right? As you see here, those are the ones you commonly know. Those are the ones most associated with the nervous system because they are the ones that do the communication, do the signaling. And then the glia cells, or neuroglia. Okay, much of these functions of the glia cells are to basically assist these neurons in their function. Okay, again, the neurons are the carriers of the information. We're going to do the electrical signaling along with the chemical signaling when we get to the target tissue. Okay, and the glia are the support. Provide physical nutrients, protections, they can speed up. We'll see there was, most of the research back in the days was on the neurons. Everybody wanted to focus on the neurons, but now they're finding out a lot of the important functions of these glia cells. Much of what the neuron and its signaling is supported or carried out by actions of these glia cells, right? So first we'll look at the structure of the neurons and the parts of the neurons that create the nerves in the nervous system, and then we'll move on to the glia cells. So first, looking at the neuron, right? This is that functional unit of the nervous system. This is the one that's going to be doing the communication. You see it's got cell body up here, and then it's going to have the long axon where it comes down into these branches, okay, and making the synapse with the target tissue. Okay, so cell body, that's the control center. That's where then the nucleus is. So all the proteins get made up in this region. So anything that has to be any proteins, they have to be shipped down these axons here, these long distances. And you got to realize that some of these axons can be several feet in length. So even though this neuron is microscopic, this neuron can be, say 3 feet long. Most of the length comes in these long branching axons. Okay. We got the regions of the cell body. We have the dendrites, little spiny processes. And so the structure of these neurons differ, but this is one of the most common neurons and called the multipolar, and that it has all these little spiny processes off here called the dendrites. Okay, this is where the neuron can receive communication. Other neurons can come in and synapse with this neuron so we can get this communication. Okay, so the neuron can receive information. It's got the cell body that's going to control, right? And then we got the neuron that is basically the region for the output signal. So input signal comes in through the dendrites or into the cell body here. And then the axon is the output signal. This is where that electrical signal is going to be carried over long distances. We're going to get that action potential. That electrical signal allows to communicate very rapidly. So again, the cell body, or also called the cell soma, it's kind of the control center. Again, it's got the nucleus, it's got all the major organelles up in this region, and it's got the cytoskeleton. It also is part of this input signal. Okay, it's part of the input. Input can come in here, but it's also part of the integration, in that the integration will take place to determine whether we're going to do an output signal, do that action potential. Okay, so that's kind of the functions the cell body serves. Now the dendrites, those are increasing the area, allows us to expand out so we can receive, because you could have thousands of neurons coming in to talk to just one single neuron. So that can take place via either the dendrites, or also neurons can come in and talk straight to the cell body. Okay, but it is the input in of the neuron. Okay, in some dendrites, okay, there is communication in a retrograde or in backwards. So even though this neuron's coming in to signal here and communicate input, there is some talk back and forth between this neuron and that neuron in some cells. And then the long extension coming off the cell body, that is the axon, that is the output region. Okay, so we're going to get integration. We're going to get input in from the dendritic end of the cell and the cell body. We're going to get integration, and then if there's enough excitatory signal, we are going to send out that electrical signal. That is going to cue the electrical signal, the action potential that allows us to transmit very quickly down this neuron. Okay, but this is the output that's taking place. Okay. This initial region, kind of the bridge between the cell body and the axon, is called the axon helix. See, it's also known as the trigger zone, the initial segment, but this is basically where integration takes place. We're going to be receiving signals here, and if there's enough excitatory signal, this is where the action potential, the electrical signal, is going to be triggered. So that is why you'll also hear this called as the trigger zone, this axon helix. Okay. Why is it called the trigger zone? Again, it's going to trigger the action potential, and what we're going to see is, I'll just bring it up here, in that we're going to find here what we call voltage-gated sodium channels. Okay, going back to your channels, your membrane transport day, what's it going to allow? It's going to allow sodium in, but only when we're at a certain membrane potential. Therefore, they are voltage gated. Okay, we're going to get into these because we're going to see this is going to be a big player in sending our electrical signal, our action potential. Okay, so second part, we're going to get into really the gory details of all this channels, the opening of these channels to cue this electrical signal. Okay, but the electrical signal is going to be sent down this axon. So the axons carry the electrical signal, and they're going to carry them down when they get to the end here. And these axons, they'll form branches. Some axons do form what called collaterals. Those were branches further up in the axon. But typically there is one axon coming down, and then it will branch into these axon terminals. And you can have hundreds to thousands of these branches here. So this one neuron can talk to many, many different tissues. Okay. At the end of these little axon terminals, you get these swellings. Okay. And these is where you're going to form the synapse with the target tissue. This is where we're going to change this electrical signal that's coming down into that chemical signal, into release of that neurotransmitter, so we can get the chemical signaling, or the cell signaling, between this neuron and, in this case, it's a neuron, but it can be many, many different tissues that a neuron is talking to. So these axons, they can travel over, they can be up to, several feet long, and they're going to be kind of the communication. They're going to be the pathways into the central nervous system and the pathways out to the central nervous system. For many of the neurons, there are interneurons that are just inside the central nervous system, but many of these peripheral neurons, the afferent and efferent neurons, they are going to be the pathways into or out of the central nervous system for integration. And so these axons, they are bundled together, and that is what makes up what you guys know as a nerve. It is a bundle of axons bundled together. Okay, you can see all these little dots as an individual axon, and they are set up to go afferent and efferent. Okay. We can have nerves that are sensory, that all the neurons bundled in there are sensory neurons, bringing information, or afferent neurons bringing information from the peripheral into the central nervous system. They can be motor or efferent, that all they do is carry all the nerves or neurons bundled in a nerve are all motor. Okay, some nerves though are moving in both directions. You have axons going one direction, axons sending information in the other direction. When going, let's say, afferent, and some going efferent, so that we can have mixed nerves. You may have learned some of these when you did your cranial nerves, because some of those are sensory, motor, and some are mixed. Now, let's classify our neurons. We can classify, we've already seen some of this terminology, in that we have sensory or afferent neurons, the ones that are carrying information from the peripheral back into the central nervous system. All right. There are interneurons. These are kind of the integrators. They lie between the sensory neurons and the motor neurons, or the efferent. Okay, but they are entirely in the central nervous system, so they don't form parts of the nerves. Okay, those will be the afferent and efferent neurons. The axons of those neurons will form the nerves. The interneurons are lined up basically in the central nervous system, in the brain and the spinal cord. Okay, they are kind of integrating neurons, and so you'll see they are usually form complex branching, in that they allow lots of integration between these neurons. Okay. And then the efferent neurons, we got the somatic, that are going out to the skeletal muscle to control muscle contraction, and we have efferents that go out to the autonomic, go out to the cardiac muscle, go out to the smooth muscle. All those organs, or all those things that are controlled without our conscious thought, are handled by the autonomic neurons. Okay, don't really need to know varicosities. We'll get into varicosities when we get into deeper into the autonomic nervous system. Okay, but realize afferent, we got the somatic going to the skeletal muscles, and the autonomic are going out to cardiac muscle, all those organs that we don't have to consciously control. So here's kind of the overall flow or connection between these neurons. We got the sensory neuron, usually some kind of receptor at this end, picks up the information or the stimulus, brings that information in. And so these sensory neurons can be visceral, okay, coming from the organs, or special senses that we will be going over in the heads, and somatosensory will also be going, or general senses that are throughout the body. Okay, those are the three components of sensory, but that all those sensory neurons bring that stimulus into the central nervous system. Okay, we can have interneurons in here, because remember the interneurons are kind of the connections between are kind of the integrators between the sensory and the motor pathways, or efferent pathways. Okay, if we need to control skeletal muscle, conscious control, we're going to go through somatic motor neurons. Okay, and then for our smooth muscle, our cardiac muscle, our glands, all those items that are controlled by the autonomic, that information can be sent through the autonomic motor neurons, or efferent pathways. Okay, and again, they're going to be either a parasympathetic or sympathetic pathway. Okay, so that's the overall flow of the information. Then next we're going to look at the glia cells, so that ends us with the neurons, and then we'll start getting into how the neurons signal. Okay, but first let's head into the glia cells, look at them, and then we'll head into the actual signaling or electrical signaling that takes place in the neurons. Alright, so our glia cells, we'll see they outnumber the neurons by quite a bit. You know, depending on the glia cell, 10 times or 50 times the amount of those cells compared to the neurons. Okay, they aren't actually physically part of the electrical signaling or transmission, but they are going to be the helpers. The neurons aren't going to be able to function properly without the aid of these glia cells. All right, so even though they aren't the components of the neuron, they are very important in making sure that the nervous system is functioning properly. Okay, provide some physical support, help in maintaining nutrients, maintaining the environment around the neurons to make sure that the signaling can take place. So here's a breakdown of the glia cells, or neuroglia. You see there are 4 glia cells we find in the central nervous system, and then there are 2 that we will find in the peripheral nervous system. We're going to go into a little more detail about the astrocytes and oligodendrocytes, so I'll talk a bit about the others here, and also the Schwann cells in detail. But in the central nervous system, we got the ependymal cells, the astrocytes, the microglia, and the oligodendrocytes. The ependymal cells, okay, and the barriers between the compartments. Remember, these cells are the ones that line those ventricles. They're going to be specialized ependymal cells that actually produce that cerebral spinal fluid that bathes those ventricles. Okay, they're also a source of neural stem cells, along with the astrocytes. The microglia, right? These are the immune cells, the resident immune cells of the central nervous system. So any pathogen comes in, hopefully the microglia there are to neutralize them and to get rid of them. Right? Then the oligodendrocytes we'll talk about in depth, but basically they are there to form the myelin, and we'll look into this in a little more detail. The oligodendrocytes form the myelin that's in the axons in the central nervous system, and the peripheral myelin is produced by Schwann cells. Okay. Along with the Schwann cells, there are satellite cells, and they serve a similar function to the astrocytes, and they provide a cushioning and try to maintain the environment for the neurons to operate properly in the peripheral nervous system. All right, so that's the basic rundown. We'll look closely here at the astrocytes and look in closer detail to the oligodendrocytes and the Schwann cells and the myelin that they produce. All right, so you can see there's quite a few functions that the astrocytes serve. They're almost like the personal assistants for the neurons to make sure they operate properly. Okay, they can release chemicals in the synapse. They provide, you imagine these neurons are very, very metabolically active, so they need substrates to make sure they can keep up with ATP production. Okay, they maintain the environment around the central nervous system. We're going to see that the environment has to be properly maintained to be able to make these electrical signals. Okay, they secrete nerve factors. They're a source of neural stem cells, and one of the other main functions is they help form the blood-brain barrier. They, along with the capillaries, the cells of the capillary beds, the endothelial, they help make this blood-brain barrier that tries to eliminate bad and foreign substances coming up to invade the central nervous system. Okay, so it's kind of a first line of defense for the brain against any kind of pathogen. Right, and you can see that here, and here are these little foot processes. These are part of the astrocytes. You saw those astrocytes are highly branching, and then they come out into these foots. Okay, along with the cells of the endothelial cells of the capillary beds. They're wrapped by these astrocytes. Okay, and so there is this interplay between the endothelial and the astrocytes in trying to help eliminate bad things coming in. Only certain things can come in. The blood-brain barrier comes in a lot with when you're talking about drugs or pharmaceuticals that are psychiatric pharmaceuticals, in that they have to reach the brain, right? Where are they going to operate? These psychiatric medicines are going to be operating on the nervous system, chiefly in the brain, right? If you make a drug that can't make its way, or pharmaceutical that can't make its way past the blood-brain barrier, it's pretty much not going to be functional, because you can't take the pill, get it into the bloodstream, and if it can't make it through here, it's not going to be able to do its job. So one of the main things in performing psychiatric medicines is they want to make sure that it's able to get in through and pass the blood-brain barrier. Okay, this eliminates a lot of stuff, but there is room for stuff to come in, and there's stuff like meningitis and so forth, so there is abilities of the pathogens to getting in through the blood-brain barrier. Okay. So the next of the neuroglia cells, the oligodendrocytes and Schwann cells. Oligodendrocytes we're going to find in the central nervous system, and Schwann cells are going to be found in the peripheral nervous system. But they serve basically the similar function in that they wrap the axons in myelin. So there are myelinated axons and unmyelinated axons. The ones that are myelinated, either they're in the central nervous system, oligodendrocytes are forming the myelin, and in the peripheral, the Schwann cells will form the myelin. They do it a little bit differently, right? The oligodendrocytes and Schwann cells, and basically that myelin, it's basically these, they wrap around and squeeze out the cytoplasm. So basically it's almost like a plasma membrane. So it's a lipid wrap, phospholipid wrap around these axons. And as you know, fat, as your professor has in his belly here, is a gun insulator. I don't get cold too often, but that insulates. Remember, we're doing electrical activity, so it's going to be able to insulate that axon. And what we're going to see later on, we're going to look at myelin and what myelin's function is to basically speed the transmission of the action potential. That electrical signal can go much rapidly if the axon is myelinated. Okay. And so the difference, again, oligodendrocytes are in the central nervous system. Schwann cells form the myelin in the peripheral nervous system. Oligodendrocytes, though, you'll see they have these extensions. Oligodendrocytes actually can wrap myelin around multiple axons in the central nervous system, where the Schwann cells, they will only wrap one axon, and we'll see multiple Schwann cells make up the myelin along the way for the peripheral nervous system with the Schwann cells, those axons in the nerves. So just looking a little closer, you can see here are kind of those concentric rings, but basically it's the plasma membrane wrapped around, and you're squeezing out the cytoplasm, so you have this concentric layers of phospholipid, almost like electrical tape wrapped around an electrical wire. Okay. Provides support, but acts as insulation for the axon. It's going to allow the speeding of transmission. And we'll see the myelin. It is not wrapped all the way down. There are gaps in between the myelin. Right? There are small unmyelinated regions of the axons along the way. And we'll see later on how this aids in the transmission, how this allows that action potential, and it will speed up the transmission of the electrical signal. Okay, these little gaps, these are called the nodes of Ranvier. Okay. All right, so we got our structure and our cells and our tissues of the nervous system out of the way. Now we're going to start looking into the functioning. How is this electrical signal taking place? What are the electrical properties of the neuron? And so, since we're talking electrical signals, okay, we're going to see nerve, muscles. These are all characterized by excitable tissues because they're able to propagate an electrical signal rapidly in response to a stimulus. Okay, so we're going to look at the electrical property, what we call the resting membrane potential of the neuron, and then we're going to look at how stimulus is formed, and then we're going to actually look how the electrical signal is propagated down the axon. And it's able to send this electrical signal over the long distance. Remember, these axons can be several feet long, so we're able to send this electrical signal down those long axons, and allows us to get long distance communication and coordination by using these neurons. So to get us started to looking at how the electrical properties of the neuron, we first want to look at what's called the resting membrane potential, or measuring what we call the membrane potential. Okay, and many cells have this membrane potential. We're going to look specifically at the neuron, but what you see is that the electrical properties, if you took a voltmeter to measure voltage and you had it in a saline bath here, similar to the cell, and you plug it in to the cell, you actually will get a reading of more negative. This is telling you that it is more negative in the cell than outside the cell. So the neuron keeps what we call a resting membrane potential. There is a difference in electrical potential outside compared to inside the cell. Okay, here again, if you have an electrode, recording electrode, and you have the reference electrode outside, you can see, right, these are the same, so there's no difference in membrane potential, so we have this nice flat line. Once you plug it into the cell, you'll get a reading of negative 70 millivolts. This is telling you it is more negative inside the cell than it is outside the cell. Okay. Well, we're going to see what makes up this resting membrane potential. If you remember what we show on our ions have charge. We see there's an unequal distribution of ions. And that's going to be one factor that maintains this negative 70 millivolts. Okay, so let's look in. There is what we call, remember, there's a chemical gradient. Or concentration gradient means there's differences in ion concentration. Here we have an electrical gradient. There's a gradient electrically inside and out. More positive outside, more negative inside. Okay. Why does this happen? Well, we're able to separate electric charges across the cell membrane. We can keep unequal distribution of ions across these cells. All right, and that is one of the main factors, is this uneven distribution of ions. And let's just bring it up now, is outside the cell. Anybody remember what outside the cell? All right, actually, let me draw my cell here. So outside the cell, we have high sodium ions. Inside, we have high potassium and low potassium outside. Okay, inside, low sodium. Remember, these have charges. So one of the main factors in maintaining this negative 70 millivolts is we have an uneven distribution of ions, with sodium high outside, low inside, potassium high inside, potassium low outside. What we're going to see is there are channels that allow potassium through, and there's channels that allow sodium. Put sodium in green. Okay. Let's just look at what way in a potassium channel, which way is potassium going to move? Yeah, you have potassium moving out. What about with sodium? Right? We're going to be using our properties of diffusion. Sodium's high outside, sodium would move in. Okay, so one thing we're going to see is, what if we had way more of these leaky potassium channels that allow potassium to leak out than we do sodium channels? So if we have a lot more potassium channels, leaky channels compared to sodium channels, that will allow more potassium to leak out rather than sodium coming in. And what's leaking out? We have more positive ion leaking out than positive ion coming into the cell. So this will be the main factor in keeping this membrane potential. Let's see how this operates. Oops. Again, factors, uneven distribution of the cell. We'll have, again, and also inside we have these large negative fixed ions, such as proteins, such as ATP. These are negative molecules inside. So again, that helps create more negative environment inside. Okay, but what we said is the differing permeability to ions. And what did we say? There were more potassium leak channels, and leak channels are open. They're not gated. They are open channels. All right. So the resting cell membrane is more permeable to potassium than sodium. Say for every sodium leak channel, there's, say, 30 potassium leak channels. That means more potassium's leaving, taking its positive charge out, than more positive charges moving in. So you can see how this helps maintain this negative environment. What other protein might be involved in this whole matter? How do we get these ion distributions, these concentration gradients of sodium? Yeah, we have to have our sodium-potassium pump. So our sodium-potassium pump's working, pump it out, but then as the pump's working, again, potassium's allowed to leak, sodium's allowed to leak in, but more potassium going out than sodium coming in. Okay. So that is what maintains the resting membrane potential. We're at -70 here. Okay. That uneven ion distribution, along with the difference in permeability of sodium potassium, keeps us at that -70. That's called our resting RMP, resting membrane potential. What we're going to see is this can change. If I change permeability of ions, I can change the membrane potential. I can move it from this -70 depending on what permeability I change for the ions. And we can see we can make, we're at resting, if I move in a more positive direction, I bring my membrane potential in a more positive direction. That is called depolarization. All right. If I've depolarized and I'm bringing the membrane potential back down to resting, that is repolarization. And then over here, where my face is sitting, you can see it goes below resting membrane potential, and that is called hyperpolarization. Let's just bring this up now so we can kind of get it out of the way and get our minds thinking around this ability to change permeability of ions can change the membrane potential. Okay, what if we had voltage gated channels, or we had channels that can open that aren't the leak channels? Okay, I have a cell and I have potassium leak channel and I have a sodium leak channel. But remember, there are a lot more of these, say 40 to just one here. Right? That maintains us at this -70 millivolt. What if I had another set of channels, a lot of channels that open to a Q, say to a neurotransmitter or to voltage? What if I had a bunch of sodium channels that I could open, or Q to open? What do you think would happen to this membrane potential? These are always open, so that's keeping us here. But what if I opened up a ton more of sodium channels? That would allow more sodium to rush in, and what does sodium bring with it? It brings with it as positive ion. All right, and that is how we can get depolarization. What if I had another set of channels, potassium channels, that open to a Q, and I could open them? I could get a membrane potential to come back down and even go to hyperpolarization, because what's happening now, besides the potassium is leaking out here, I opened up a ton more channels where more potassium can leak out, and so what's leaving? There's more positive charge leaving, and I can bring my membrane potential back down. So this is what we're going to be doing to make our electrical currents to make our electrical signal. We can depolarize, repolarize. We can change the membrane potential to send our electrical signal. So rushing in, ions, the movement of charge is basically your electrical signal. We always think of it as electrons, right? Negative electrons moving through a wire. That's our electricity. Here, we're going to be moving positive ions down the axon to give us an electrical signal. We are moving charge. Okay? So let's look at how this is set up again with our separation of electrical charge. A cell membrane separates it, allows us to create an electrical gradient. Difference in that charge, remember, more negative inside than outside. We also have a concentration gradient because we have difference of ions from inside to outside. Okay, if we put these two together, we can say there is an electrical chemical gradient. Okay, there are two factors in play, and these two factors are what going to determine how my ions move. Okay, I can move ions due to the concentration gradient, but we're also going to see that the electrical gradient can also have an effect on the ion movements. Okay. One thing to keep in mind is positive is attracted to negative charge, right? And positive is repelled by positive. Okay, so keep that in mind as we start talking about how the electrical gradient comes into play on these movement of ions. And just to keep in mind, again, there's an electrical gradient. There's a difference in charge between the ECF and the ICF. So let's take a closer look at our differences in ions, our chemical disequilibrium from inside and outside the cell. Two columns we're really looking at are the I is actually the extracellular, the ECF, I for interstitial, so the ECF, and C is cellular, so the ICF, intracellular. And you can see exactly what we're talking about with the differences in here: sodium, high outside, low inside, and the opposite end, potassium, low outside, high inside. And you see the differences for several other ions, the chloride ions and calcium. They are both high outside, low inside, so they are similar in effect to the sodium. All right, so as I said, there are two forces that are going to be in play in what is how the ions move. Okay. For ion diffusion, we've already seen diffusion, right? We've seen diffusion. Concentration gradients are going to cause molecules to move, but now we're adding another component in here. There is a electrical gradient that is also a force that can have an effect on movement of ions. Okay, so we have a chemical gradient and an electrical gradient, and so we have an electrical chemical gradient, and we're going to see these ions can move due to this electrical force and chemical force are both going to have an influence on the movement of ions, and it's going to allow us to figure out what's called the electrical chemical equilibrium. Okay. When both forces are balanced, then the ions are said to be in electrochemical equilibrium. Okay. Let's see how this operates. Uh-oh. This is where we do have a little mathematics in this course. Okay. This E is for the electrical chemical equilibrium. The ion could be, in our case, what the main ions we're looking at. It could be sodium, or it could be, say, potassium. All right. What the electrical chemical equilibrium is going to tell you is that what membrane potential, okay, what is the membrane potential from inside compared to outside? At what membrane potential, what millivoltage will the ions stop moving? Okay, so the equation is E ion, depending on what ion. Z is the charge of the ion, so let's just say we're dealing with sodium. That would be 1, a positive 1. Okay. We'll have the ions, I'll just, okay, it's going to be high outside compared to inside for sodium, and we'll see we'll get a positive number. It'll be somewhere around positive 60. Okay, because this is a positive number here. If we had potassium, on the other hand, it would be low up here and high up here, or high down there. Okay, this is what we call the Nernst equation. It predicts at what membrane potential will the two forces equal. We have a force, a concentration, a chemical force. The concentration gradient is telling the molecule to move in one direction. At one point, the electrical force is going to be going in the opposite direction. And when those forces that are moving, forcing, say, the molecule out, and this force is bringing it in, when these forces equal, that is when the ions will stop diffusing. They will be in electrical chemical equilibrium. All right, let's see how this operates. Okay, when we're using the Nernst equation, we're just looking at what if the membrane was permeable to only one ion. Right, we're not looking at effects of both, we're just looking at what if the membrane was only permeable to one specific ion. Okay. At what point does the membrane potential oppose the force of the concentration gradient? That is when we are at electrical chemical equilibrium. And that is what the Nernst equation is predicting. Okay. Let's look at our friend potassium. If we use the Nernst equation, we got E61 because it's a positive, right? There's the Z under here, but it's 1, so it's 61. And log, what's the log? We got almost 30 times more potassium inside than we do outside. If you actually plug this number in, negative and negative 90 millivolts. All right. What this is telling you is that when the membrane potential reaches negative 90 millivolts, more negative inside than outside, the potassium will stop moving. The forces, the concentration gradients saying go out potassium, but at a certain point the electrical force is trying to pull potassium back in. When those forces equal, potassium will stop moving, diffusing. Let's look at our cell. So we're at negative 70 millivolts. Okay, what the nurse is saying is that if we were permeable to potassium, remember potassium is high inside and low outside. Okay. If I'm permeable to potassium, which way is potassium going to go? The concentration gradient is saying move out. As potassium moves out, what happens to this number here? Potassium is taking out its positive. So this number starts becoming more negative. And let's say we reach so much potassium is moved out that we're at negative 90 millivolts. Remember, potassium, there's an electrical, so the concentration gradient is saying potassium, go out. Electrically, though, what is potassium? Potassium is a positive. And what's happening to inside the cell? It's becoming more negative. Will negative attract positive? Yeah, at a certain point, that force, that negative force is attracted to that positive potassium. There's a force saying, hey, I want you to stay here. Positive is attracted to that negative membrane potential. As it becomes more and more negative, the force becomes greater. At a certain point, these two forces, the concentration gradient saying going this way, the electrical force is saying stay in, and when those two forces equal, potassium will not diffuse in or out. Okay, and so that is what the Nernst equation is telling you. At what membrane potential are those two forces equal? The concentration force and the electrical force. Chemical force and the electrical force. For potassium, and these are the physiological numbers in the body, and this is typically what the concentration would be outside the cell and what the concentration would be inside the cell. So if we were just permeable to potassium, it would move all the way till it reached negative 90 millivolts. Okay. What about our friend sodium? Okay, let's look at its equilibrium potential. We got almost the opposite. We got 61 over 1 because sodium is a positive 1. Log is more outside than inside, 10 times more outside than inside. We actually come up with 6 to the 10, around 61 millivolts. Okay. Look at sodium. Sodium on the other, if we were, remember the nurse is just saying we're only, hypothetically, we are only permeable to the sodium. Sodium is high outside and low inside. Okay, we're permeable to sodium. Which way is sodium going to move? The chemical gradient is saying move inside. That's the chemical force. Okay? As sodium keeps rushing in, at a certain point, the membrane potential starts turning positive. Okay? What happens when positive charge is trying to move in against a more positive environment? They're going to repel each other. The electrical force is saying, repelling you. It's the inside's becoming more positive. It's starting to have a repelling force. The electrical force is saying, no, stay out, where the concentration, the chemical gradient for sodium is saying, come in. All right? At what membrane potential are those two forces equal? All right, and what the nurse is telling you is that at 61 millivolts, the electrical force is equal to the chemical force, and sodium is at equilibrium, at the electrical chemical equilibrium. Okay, that's all the nurse is telling you. Why do we go through all this? Where does our potassium? Our potassium was, electrical chemical equilibrium was at negative 90 millivolts. Where does our resting membrane potential sit? At negative 70 millivolts. Okay, this is potassium. Where's our sodium electrical chemical equilibrium? At positive 60 millivolts. Okay. Where does my resting membrane sit? Where does it sit closer to? Yeah, it definitely sits a lot closer to the potassium equilibrium. So what this is telling you, what do we say about the permeability of potassium at rest? There's more permeability to potassium than there is to sodium, and henceforth, this is why our membrane potential is sitting at negative 70, not closer to positive 60. Okay, due to the leakiness to potassium. The cell at rest, the cell is more leaky to potassium than it is to sodium coming in, and therefore our membrane potential is much closer to the potassium electrochemical equilibrium. Right? Here's some numbers looking at, okay, you can see what calcium, chloride, they're similar to sodium in that they would move in. The difference is chloride would be bringing in a negative charge, and then calcium would bring in a positive 2 charge. We'll be seeing these guys a little bit, but we're going to be focused mostly on these ions, because these are the ones that are going to run our action potential, our sodium and our potassium. And I'm just going to beat it down some more, though, is resting calcium channel, the leak channels. We are more leaky to potassium than we are to sodium, and therefore that is why we're at that negative 70 millivolt. This is actually a pretty good diagram in that what makes or cause of the resting membrane potential. Okay, the sodium potassium pump's always working, keeping this higher on inside and this higher outside, but we have those fixed ions, but the unequal permeability is what is the main draw to keeping us at the negative 70. We are more permeable to potassium and less permeable to sodium when at rest. Okay. Ion gradients, leakiness. But what would happen if the potassium pump stopped working? Would we be able to create this gradient and allow for, at a certain point, the potassium, if we didn't have the sodium-potassium pump, those ions would come into equilibrium and we wouldn't be leaking potassium and so forth, and we wouldn't have our gradient. So the sodium-potassium pump's not actually the main factor in doing it, but it's the one that keeps the ions in disequilibrium to allow for the leakiness to cause for our membrane potential. So how we're going to go about sending electrical signals is by changing the membrane potential. We're going to be changing it from that -70, either moving it more positive or more negative. And the way we're going to do this is we're able to do it with gated ion channels. Okay, we see the resting membrane potential. The resting membrane potential is set up by those leaky channels. Leaky channels are non-gated. They are always open, so they're always allowing potassium out and sodium in. Key is, remember, more potassium channels, non-gated potassium channels, leak channels, compared to sodium. So more potassium leaves compared to sodium coming in. That is at rest. Okay, we're going to be able to open up gated ion channels, and that's going to allow us to allow either more sodium in or more potassium out, and that can change our membrane potential. Okay, move it away from that -70, move it away from that resting membrane potential. Okay, like I said, non-gated, more of these, potassium compared to sodium leak channels, but they're always open, they're always allowing them to leak out. Whereas our gated channels, we can open our gated channels mechanically. Okay, we're going to see this later on. We're going to see it more with the sensory nervous system. Okay, we can open up, say if we mechanically open up more sodium channels, what do you think would happen? Here's our resting membrane potential. It would go more positive and a depolarization. All right. We can also have chemically gated. That responds to neurotransmitter. If I had a neurotransmitter that say open a sodium channel, again, what would happen? Resting membrane potential, I open up more sodium channels, I would get depolarization. Right? What if I was moving and I opened up more potassium channels? More potassium allowed to leave, I would get hyperpolarization. Okay, so we're going to be able to move. Right, this is -70 millivolts. Move this membrane potential either more positive or more negative by changing the permeability to these ions by opening up another set of channels. Okay, we'll also see voltage gated changes that respond to changes in the membrane potential. Okay, this is what's going, these voltage gated channels are what's going to be the main player in allowing our action potential to take place. Okay, but what opens them is we get a change in membrane potential. All right, what if my membrane potential becomes more positive? That could open up some voltage gated channels and allow more ions through. Okay, so by changing the amount of channels open for a certain ion, changes that permeability to the ion and can change my membrane potential either up or down depending on what ion channel is being open. Okay, so how are we going to send our electrical signal? We're going to change permeability of the ion. If we change permeability, allowing ions to move in and flow, we're moving charge. If we move charge, that is an electrical signal. All right, we're not moving electrons in this case. The charges that are moving are the ion charges. Okay, those of sodium moving in and moving down to create an electrical signal. All right, so depending on what ion we open to, we can either depolarize or hyperpolarize the cell. Okay, let's say it's sodium. Let's say we got potassium channel. Okay, this is ECF. ECF. Let's just look at changing flow of ions. If I opened up more sodium channels, which way is sodium going to go? Sodium is high in the ECF, so sodium would come in. All right, what do you think would change in my membrane potential for my sodium if I opened up more sodium channels? Negative 70 millivolts. Okay, here I open up sodium channels. More positive ion is coming in the cell, and that's going to cause my depolarization. Now potassium, where's potassium? Potassium is high inside. So if I opened up more, I have gated potassium channels that I open up, more potassium is going to leave than say what's happening at rest. What's potassium taking with it? It's positive charge. So if I was measuring membrane potential, it's at negative 70 millivolts at rest, right? Coming. Now I open up I open up these gated potassium channels. What would happen? More positive is leaving, and therefore my membrane potential would go in hyperpolarization, go in a more negative direction. Okay, so this is what we're going to be doing. We can open up these different sets of channels, change whether more or less sodium or potassium's either leaving or sodium's coming in, and that's going to change my membrane potential. All right, that's all we're doing is opening up these other sets of channels to get either more positive ions coming in or more positive ions leaving, and that's going to change my membrane potential. And so looking at the different electrical signals in the neuron, there are two types of electrical signals. There are the graded potentials, and these are going to happen. They're over short distances. They are graded, so they are variable. I can have a graded potential that is small, or I can have a graded potential that is really big. Okay, they are variable in strength, big and small. Okay, we will see they lose strength as they travel across the neuron. The graded potentials are going to happen at the dendrites, at the cell body. The graded potentials are ultimately what's going to, if there's enough excitatory graded potentials, it's going to cue the action potential that we see takes place down at the axon. Okay? An action potential, unlike a graded, is very brief. It is, I measured at one point of the axon, it would be this, and I measure again in another part of the axon, right? What's the difference? The graded potentials, I could have small or big. There is no difference in the size of my action potential, and also as say this is at the beginning of the axon and this is at the end, what don't you see happening? It's not losing strength. Okay, I know it looks a little funny here with my sad drawings, but we'll show you some pictures and show you some diagrams to get you clear on what I'm talking about here, the difference between graded and losing strength, and the action potential being all or none and also not losing strength along the way. Okay, but we're going to start with the graded potentials because this is going to be the cue to get us to our action potentials. Okay, remember these are happening at the input end. They are happening at the cell body and the dendrite. Okay, these changes in potential. And what to cue our action potential, what we're going to need is a large enough depolarization to cue our action potential. Alright, so we're going to start with the graded potentials. Remember, the graded potentials are taking place. Again, potential means we're changing the membrane potential. But these graded potentials are taking place either at the dendrites or the cell bodies. And they can take place, depending, it can be, we're going to see later on with sensory system, what causes graded potentials are some kind of stimuli. But let's just say, in this case, it's a neurotransmitter that's causing this graded potential. Okay, say we have a chemically gated channels. The neurotransmitter is going to open up a certain ion channel, a ligand-gated ion channel. Okay, judging by what we're measuring here, we see we're at resting membrane potential, but we're going up in a more positive direction. Let's just quiz you. What kind of ion channel do you think is being open here? A sodium ion channel or a potassium? What direction are we going? We're going more positive. So that means we have to bring more positive ions into the cell rather than letting more positive ions leave. And how will we do that? Right? Your best bet here would be a ligand-gated sodium channel that allows more sodium to come in, and that would cause the depolarization. Okay? Let's just watch though as we're going here, because where we're going to cue action potentials, if you remember, we're going to cue them at the trigger zone. Okay? And the magic number we have to get at the trigger zone is we have to have a membrane potential of, say, negative 55 millivolts. Okay, here's that negative 55. Trying to reach negative 55 here because that's going to be the cue for our action potential. Remember, our graded potentials are going to be the cue to ultimately send that long distance electrical signal down the axon. Okay. Two things about graded potentials. They can be graded, they can be large, or they can be small. All right, the strength, that's, we're talking about the size here. But what's also different from the graded potentials compared to the action potential is as they travel through here, through the cell, they're meeting resistance by the cytoplasm, and what we're also going to see is the electrical signal can leak out. So as it travels, you can see that the signal is actually diminishing. Okay, the original stimulus is happening here. You can see how high, or the difference in the change of membrane potential is close to like from negative 70 to negative 40. We went in a positive 30 direction. But as that signal travels through, it loses, I like to say loses steam, and that when we get there and we measure it, it is not that much change. It's not like here, down here. Okay, so this is showing you that these graded potentials, as they travel through the cell, are losing steam. Okay, they're meeting with resistance, and so they lose strength as they travel through the cell. Okay. And what we can see is these can be depolarizations, they can be excitatory, or they could be inhibitory. Let's go back and let's say we opened up a potassium channel here. Do you think we would be depolarization? No. The signal would be more of a hyperpolarization. Okay, hyperpolarizing is inhibitory, in that it would inhibit the neuron from firing. It would inhibit it from making an action potential. Okay, because remember, where do we want to be for an action potential to be triggered? We got to be at negative 55. That is in a more positive direction than the negative 70 millivolts. Right? So our depolarizations are excitatory because they push us closer to that negative 55 millivolts, that magic number we got to reach here at the axon HELOC. Okay, so depolarizing graded potentials are excitatory, and hyperpolarizing are inhibitory. Okay, let's look at, we're measuring here at the trigger zone at the action potential. Remember, we have to get to that magic number of negative 55 to trigger an action potential to take place. Here we're above negative 55, but what happens as the signal diminishes, by the time we reach the trigger zone, we're below negative 55. So there is no action potential. We haven't reached this negative 55 is called threshold. Threshold for the action potential to take place. And as I said, in this case, likely it's a chemically gated channel that's being open. In this case, it's a depolarization. Likelihood it is a sodium channel. We're going to see mechanically gated channels when we go to sensory, right? And then when we talk here, what's causing our action potential, because our action potential is a strong depolarization with the repolarization, hyperpolarization, we're going to see there are voltage gated channels down here. All right, but let's get us to a graded potential that actually cues our action potential. Okay, before we get there though, let's look at graded potentials. You can see what is taking place here. Remember, we say as we move through the cell, here's the stimulus, the sodium's coming in. But as the sodium ions move through, okay, the graded potential decreases. You see it's big here, but it's small as it moves through, right? And why do we lose strength? There is cytoplasmic resistance as we move through the cell, and you can also see some of the signal also leaks, right? This is why these graded potentials decrease or decremental as they move through the cell body, from the dendrites into the cell body down towards the trigger zone, down towards that axon HELOC. Again, just showing more of the local current and how the flow diminishes as we move through the cell. There's current leaking, and there's resistance as we move away from the initial stimulus, the initial change in membrane potential. Alright, so this is where we've seen, remember, our graded potentials trigger the action potentials. What's the magic number? We got to reach negative 55. We got to reach what's called threshold for the action potential. Here is that one we saw originally. The stimulus is above, right? It's above threshold. It's above negative 55 at the initial, but as we reach the trigger zone, we're called subthreshold. We're below threshold, and so there is no action potential triggered. But let's say we have a bigger stimulus. Okay, in this case, let's just say there's bigger stimulus, there's more sodium channels that are open. We're going to have a larger depolarization. Okay, you can see how this depolarization here is much larger than this one. And as we move through, it decreases, but by the time we reach trigger zone, we're still above threshold. We're still above negative 55. We're above threshold. And what happens is we get the triggering of the action potential because we are above threshold. Okay, so our graded potentials, depending on size. And what you got to recognize too is that this is oversimplified and there's just one neuron talking to here. There can be hundreds to thousands of neurons talking to this one neuron, sending either excitatory or inhibitory signals, and they can be summed together as long as there's enough excitatory signal to get us to negative 55 here. We're going to trigger an action potential. Okay, we're going to trigger, boom, and we're going to see the action potential is all or none. Either it's going or it's not. There's no in between for the action potential. Either it's light switches on or light switches off. Here, light switch is on, we triggered it. Here, the light switch is off because there's no action potential triggered. So we've talked about how the graded potentials trigger an action potential at the trigger zone. So what magic is taking place at the trigger zone? Okay, what we see is, why do we need to reach this negative 55 millivolts? Right? Why do we need to reach that here at the trigger zone? Okay. Well, the key is that the change in membrane potential to make our action potential take place is going to be triggered by channels for sodium and channels for potassium. They are going to be the carriers of our electrical current, our action potential down our axon. But what cues is these to open, the channels here on the axon, is they are voltage gated. Okay, and what cues them is the change in membrane potential. Negative 55 millivolts are going to cue these channels to open. And chiefly at the trigger zone, as we have a high concentration of voltage-gated sodium channels. Right, and what we're going to see is we hit -55, those voltage-gated channels open. Now a ton of sodium can come in, and we're going to get this large depolarization that cues our first start of our action potential, this electrical signal that's going to travel down the axon. All right, so that is what is happening. Those graded potentials, they reach -55 here. That is going to cue the action potential at the trigger zone because it's going to open those sets of voltage-gated sodium channels that are located right there. Okay, so that's the magic, is the high concentration of voltage-gated sodium channels at the trigger zone. Okay, we already discussed a little bit of graded potentials and opening channels, but we can get excitatory postsynaptic potential. Excitatory in that it is depolarization. We can have inhibitory postsynaptic potential, and those would be graded potentials that cause hyperpolarization. Okay, these would be, we saw simplification of the graded potentials in that we just had one stimulus coming in. But remember, these neurons, they could be talked to by hundreds to thousands of other neurons. So they're going to be getting signals, excitatory and inhibitory, from many different neurons. And so what we can see is these excitatory signals and inhibitory signals can be summed together. Right? If the sum is there is enough excitatory potential, and if there's enough excitatory potential, we can probably reach that -55, that threshold at the axon HELOC. So let's see how we get some summing. Okay, we can get what's called spatial summation. Okay, here's multiple neurons. If these multiple neurons are signaling almost simultaneously or within short time of each other, okay, they can move through the cell at roughly the same time, and those signals can be summed together. They are over space because they are coming from multiple neurons, and if enough of this is excitatory and there's a big enough signal, when it reaches the trigger zone, we can cue that action potential. Okay, spatial, over space, because they're coming from different locations, different neurons. Now, temporal summation. Okay, what if we had one stimulus coming in, but it was coming in, hey, we got a stimulus from the same location, and then a little bit later stimulus. There's no summation. But what if that one neuron signal back to back, boom, boom, we can get what's called temporal summation over time. Okay. So two subthreshold graded potentials, but from the same presynaptic neuron. It's just that they are so close in time that they can have an added effect to each other, and if they're strong enough at the axon HELOC, they can reach threshold to initiate. This is our action potential here. Okay, so spatial over space, multiple neurons speaking at a similar time, where temporal, it's boom, boom. The same neuron is firing so quickly that those excitatory signals can be summed together. All right. And ultimately, those graded potentials are going to cue, right? If we reach that negative 55, we reach the threshold here, it's going to cue our action potential. This is what our action potential will look like. And look as we measure down. Remember, the difference between action potential and graded potential was graded can be, we can have big graded potentials, or we can have small graded potentials, or even medium graded potentials. What do you see with the action potential? If you measure an action potential all the way down, they are all of the same strength. They are not graded. Also, what's different between graded potential? Graded potentials, if this was a graded potential, not an action potential, if we measured here, if we measured down here, if it was a graded potential, it would be much smaller because it would lose steam. Okay, it would lose strength. What we see with the action potential is we don't lose strength. Okay, and we'll see why this takes place. But as we're moving, this will come to the end of the part one. We'll move into the gory details of what's taking place in this action potential. But what you can see is there is this is one, we have a large depolarization. So think opening of sodium channels along with a repolarization. Think potassium channels as we move forward. Again, what kind of channels are these? These are going to be voltage gated sodium and potassium channels. Okay, so we will get into the gory details of this in part two, and then we'll once we bring this signal, we bring this action potential all the way down, it'll bring us to the axon terminal, and we can get into the neurotransmitter release and the chemical signaling. Okay, but first we'll focus on how do we do what we call propagate this action potential down the axon.